Sorry for being a little bit late, but the lecture file for Zoom was running on my computer. What it is it's making the video that I can post online from the Zoom lesson. Anyways, that was about to end, so I wanted to wait until it ended because my computer works much faster when that Zoom file isn't running. So I waited to that to end, and that took like an extra 2 minutes. Anyways, are there any questions before we begin? If not, let's go ahead and talk about what we're going to be doing. Today, we're going to be doing Lab 01, the ubiquity of microorganisms. Remember that you need to turn in Lab 00 by 8 a.m. Friday, January 12th. If that is not in, and then I go and look to see who is attending this class, if you do not have worksheet for Lab 00 in, I will drop you from the class. You should also read about the read about the infectious disease research project. You can find that under the project instructions on Canvas. Let me show you. Oh, come on. I can't get it to open up. Why is that not the case? For some reason, this is shut. There we go. Leave it open. All right. So under week 2 lab, there is the infectious disease research project. I'm going to be talking about that next week. So you should look at that and have it read by next week. Any questions about any of that? Nope, not there. I want this. Anyways, that's a project that we'll be working on for a large number of weeks, but I'm going to be talking about it next week. Oh, and then one other thing I want to remind you, you do need to take the online plagiarism quiz. I don't think that's right. I think it's 11:59 p.m. on Saturday. Let me check that. Until 11:59 p.m. January 13th. So I need to change then the syllabus. That's not right. And I'll update that later on the web page. I make a note that I need to update that. All right. Any questions about what we're going to do? We'll be discussing Lab 01, ubiquity of microorganisms. And this lab must be due by Saturday, 11:59 p.m. this Saturday. Here. Nope. Module 1, Ubiquity of Microorganisms. There is some reading for all of the labs that must be done before you work on the lab module. So please read, not very much, but please read in Tutoria, page 157 to 162 on culture media, and page 162, 63 on obtaining pure cultures. So that's one page, 3 or 4 pages. There are also some video clips in this lab module. The learning objectives: know that microorganisms are almost everywhere. Describe the purpose of culture media. 3, describe what colony abundance is and what morphology is. Colony morphology I'm talking about. And 4, be able to define the following terms: inoculation, ubiquity of microorganisms, what's something that's sterile, isolation, what's isolation, incubation, in vitro, like in vitro growth, culture media. And of culture media, you should know the different types. We have a broth culture media, which is a liquid media. And then we have an agar culture media, which is a solid culture media. And then we can grow the solids in a slant, in a tube, or a deep. And I think I'll show those later. And then what's something that's fastidious? What's a non-selective media? What does ubiquitous mean? And what is a colony, a bacterial colony? Microorganisms are organisms that are too small to be seen with the naked eye. This does include viruses and prions, although they are not organisms. And they are in this group, even though they're not exactly alive and they're not cellular. We'll also include helminths, worms in our studies, even though the adults are not usually microscopic. However, their infective stages of the worms are microscopic. The term ubiquity of microorganism refers to the concept that microorganisms are essentially everywhere. And if that's the case, then that's a problem for growing things in a real microbiology lab, because we need to grow our microbes in a sterile environment. Sterile means it's free of living cells. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn basic lab protocols. You should know that a culture medium, also called a growth medium, is a sterile laboratory preparation that contains a variety of nutrients that are required for growing microbes in the lab. A culture medium may be a liquid. These we call broths, and they are commonly used in test tubes. We do add agar to the liquid medium to result in a solid medium. You could add something else, but most of the time we add agar. You could add jello, you could add starch, but most of the times we add agar. Here's a little video for you to watch on preparing pour plates. You can watch it after I'm done talking. Note how the culture medium needs to be sterilized prior to its use. Because if you're to try to grow something in a culture medium and it's not sterile, you're just going to have all sorts of organisms growing in it. An important question you must consider is, why must a culture medium be sterile prior to it being used? Here we have different culture media, and this is a liquid broth. This is a slant, which is a solid culture media containing agar. So that'd be the same media, but just has agar added, making it solid. And we put this one in a slant. This is called a deep. It is an agar solid medium, and you just let it go across flatwise. And those are Petri dishes containing solid media, agar plates, different types of media. There are a variety of different media microbiologists can use. Today, we're only going to talk about one, the general purpose media. A general purpose media is non-selective in function. It's usually a complex composition made of many different materials, and the general purpose media support the growth of most non-fastidious organisms, meaning you can get many things to grow in a general purpose media, but you cannot get a fastidious organism to grow in a general purpose media. A fastidious organism is one which requires certain additives to be added to the media before the organism will grow. And today we're not going to talk much about fastidious cells, but Streptococcus pyogenes, which causes strep throat and a whole bunch of other human diseases, is a fastidious organism. We cannot get it to grow on a general purpose media. Some examples of a general purpose media include nutrient agar with yeast extract, abbreviated NA for nutrient agar for yeast extract, tryptic soy agar, abbreviated TSA, and then nutrient agar, or abbreviated NA. These two are the same. It's just that this has yeast extract added, so this one's a little richer than nutrient agar. Whenever you're working in the microbiology lab, you should label all your Petri plates and your tubes correctly with the following 4 or 5 items. They all must have your name, the date, the type of medium. So if you're growing it on a nutrient agar with yeast extract plate, you should put NA on it, because nutrient agar with yeast extract would be rather long to write. Inoculum, you should talk about what's growing in the culture. Like if you're growing E. coli, you write E. coli. If it's something like a plate that's been exposed to the air, you write air exposure. And then the incubation temperature is optional. It can be written on the plate, but it's not required. Usually you write on a test tube by writing on the tape and then sticking the tape to the test tube. On a Petri dish, you usually write on the bottom of the plate, on the edge of the bottom. We don't want to write in this area of the plate, because if you do, you won't be able to clearly view the organisms in the plate. So you move it off to the side. So that's the instructor's name. There's the name of what's growing on the plate. This is the date. This is the media. And then that's the temperature. Remember, the temperature that you grow it at is optional. Inoculation is the placement or transfer of a microbial specimen into or onto a sterile culture medium. So we inoculate the tube by adding microbes to it. Incubation is the placement of the culture media in an appropriate physical environment for growing it for a prescribed period of time. And this is just to allow the growth of the microbes. Usually, we grow our microbes in an incubator. Our incubators are rather simple. All they do is they add temperature and a little bit of humidity to grow the microbes. However, you can have more complicated incubators, which could include an oxygen level, carbon dioxide level, as well as the temperature level, the humidity, And then, of course, the incubator, you grow them for a specific time. Let me show you an incubator. Here's a fairly complex one that can set different dials, probably for the amount of oxygen and carbon dioxide, as well as the temperature. And you notice this container here, what we do is we put water in there and then shut the door, and this warms it up, causing humidity to occur in the incubator. Most incubators simply add water to the incubator in a dish, although there are very complicated incubators which you could set the humidity level with the dials, and it would add the control the humidity within a specific range, at least while the door is shut. All right, you should note that during incubation, when you have an agar plate in the incubator, the warmth of the incubator will cause the water from the agar to come out, evaporate from the plate, and then condense on the lid of the plate. And then the drop of water will form a drop and then drop into the plate, which would disperse the microorganisms. It does not contaminate the plate, although it does add risk that there may be more contamination just because you're getting the water dropping from the lid. If the water drop were to come down the edge and then come into the plate, that could contaminate the plate. That likely would contaminate the plate because the edge right here is not sterile. It is sterile on the lid though. And so if the drop comes down, most of the time it will not contaminate the plate. So to prevent this dropping of water from the lid onto the culture, which would then spread the cultures on the plate, what we do is we incubate our plates upside down in the incubator. Then, if water were to come out of the agar, it would condense on the top, which is going to be the agar, and then the agar will wick the moisture throughout the agar. But if a drop of water were to form, and it shouldn't, it would drop on the lid, and the bacteria isn't grown on the lid, so we don't care if water drops on an upside down plate because it'd drop on the lid and there's no growth there. Anyways, the important part is to know that all plates, when grown in the incubator, should be put in the incubator upside down to prevent the condensation of water from occurring on the lid and then forming a drop, which will then drop down onto the growth. When we have colonies growing in the agar plate, they will form visible bumps on the agar surface. Each of these is a colony. Bacterial colonies tend to have rather smooth textured surfaces. Fungal colonies tend to be furry or hairy looking. Let me show you the difference. This is a fungal colony, kind of hairy looking, and that one is furry or hairy looking, also a fungal colony. These right here are bacteria colonies, and you notice how they have smooth and non-hairy edges. Each colony is made up of tens to hundreds of millions of cells, all descendants potentially of one cell that was placed there during inoculation. That one cell splits into two, two into four, four into eight, eight into 16, 16 into 32, etc., until you get about 1 million cells And if you have 1,000,000 cells, you will have a very tiny colony that you can see. And it could be, most colonies are tens to maybe even hundreds of millions of cells. And they're all producing by binary fission, where one cell splits into two. Here's a little video for you to watch on binary fission. So short, I probably should watch it together, but I'll let you guys do that. In theory, one cell leads to the formation of one colony, yet a colony may be formed by more than one cell. A colony could be formed by two cells or by a clump of cells. For example, here we got a streak, and you notice there's some of different sizes. These colonies are all pretty small. Each colony may be formed by one cell. This colony is pretty large. This colony was probably formed by at least two cells, or maybe a clump of cells. And that's why it's much bigger than the others. Any question about that? I'm talking about these colonies because they're the same species. That one and that one are obviously different species. All right, colonies are not visible in a broth culture, and that's because when things are growing in a liquid broth, all of the cells just mix together in the liquid. There are no colonies in a broth culture. There are some obvious visible colony morphological variations between different species of bacteria, and that's why that looks different than that. And then these two are probably the same species, why they look similar. And that one and this one are also different than those. So you can use those differences to characterize different colonies and say that one colony is one species and another colony is another species. There are different characteristics you can use. You can take the shape of the colony. You can take the margin. The margin is the edge of the colony. Entire means it has a smooth edge, undulate, lobate, and curled, a rough edge, rhizoid, and filamentous, either a rhizoid or filamentous edge. Whenever I'm asking a question about a colony, I would not be expecting you to differentiate between rhizoid and filamentous and tell me this colony is rhizoid, that is filamentous. I would expect you to be able to tell me the difference between an entire margin and either rhizoid or filamentous. And likewise, I would tell you, you don't need to separate undulate and lobate and curled from each other because they're all similar, but you should be able to separate entire from undulate, lobate, or curled. Elevation is the elevation of the colony. The colony could be flat, raised, convex, fulvinate, that's extreme convex, and umbinate, mountain shape. I would not expect you to distinguish between a convex colony and a fulvinate colony. You can distinguish between the size of a colony. There's punctiform, which is a pinprick. There's small. Small is on the size of a dull pencil point or a pen point. Small. Moderate is in between the pencil point and the eraser side of the pencil. Anything that's large is bigger than the eraser side of a pencil. You can distinguish a colony by smooth or rough, whether the colony is glistening or shiny, or whether it is dull, whether the colony is pigmented, meaning it has a color, or it's non-pigmented, it lacks color. And non-pigmented could be white, but it could be cream colored, off-white, or tan colored. The thing is, all of those are non-colored. And then lastly, there's the optical property of the colony. Most colonies are opaque. You cannot see through them. Some of them are translucent, that you can see partially through them, sort of like slightly dirty water, slightly muddy water. You still could see the bottom of the stream, but the stream is muddy, so you can't see the bottom clearly. And then transparent, very few colonies are transparent. Transparent would be like liquid water is transparent. You can see through it. Let me see if I can find anything there. Not there. There we go. These colonies are opaque. This colony is translucent. That colony, the big one, is translucent. That one may be opaque. But like I said, translucent, you can partially see through them. Maybe best example is here. You partially see through this, but you can't see through that one, so that's opaque. And that's probably two colonies growing on top of each other. Any question about any of that? All right, let's talk about some of these. Colony A is a large colony, pale yellow in color. I would call that probably off-white. Circular in shape, maybe. Shiny, yeah, right there it's shiny. Oh, fulvinate, that one we can't tell. And then the entire edge. Colony B is medium in size. Actually, I would say that's probably large, but I'm not sure how big this plate is looking. If that's the regular size of a plate, that would be a large colony, because that would be larger than an eraser. Cream colored, so non-pigmented, irregular shape, I'd agree with that. And then that's a rough colony, and you can't tell whether it's flat or not. Colony C, a small colony. White in color, circular shape. It doesn't look like it's perfectly circular, but we'll go with it. A smooth colony, and we can't tell the convex and entire margin. To determine the elevation, what you really need to do is to take off the lid of the plate and then look at the colony from the side. And then you can tell the elevation. When you're looking at the top, from on top, it's very hard to determine the elevation. Like I cannot tell this one, colony A, is pulvinate. I can't tell that from there when you're looking at the top. And that says flat. I can't tell that either. Convex, colony C. Like I said, these all look flat to me, but that's because we're looking at them from on top. You need to, for the elevation, you need to look at it from the side. All right, when we're talking about colonies, it's important to know the term abundance is simply referring to the total number of colonies on an agar plate. So that's a bit too many, but if we go back here, the total abundance, that's too many too. Let me see if I have a better picture. Nope, I don't have a better picture. Here we can talk about the abundance of cats. This isn't This isn't colonies, but the abundance of cats are 1, 2, 3, 4, 5, 6, 7. So the abundance is 7 wild cats. The diversity of cat species is 2 species, the lions and cheetahs. So the abundance is the total number of colonies. The diversity is the number of different kinds of colonies. And to get the diversity, you look at the colony characteristics, like the color, the shape, the texture, to identify the different colony types. Let's come up to one that's easy. All right, all of these, as well as these, and most of these, are all one colony type. That would be a second colony type. This would be a third colony type, a fourth colony type, probably a fifth colony type, and then a sixth colony type and a seventh colony type. So on this plate, the diversity is at least 7. Any questions about that? And if that's a different one than that, or different than that, you might have more than 7, but it's at least 7 on this plate. And then the abundance would be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. The abundance is going to be 40 to 50 colonies. Okay, you can actually do the exact count. I didn't want to spend the time doing the exact count. When I ask you for the abundance, you just have to be approximately correct. With the diversity, you also have to be approximately correct. However, if you're way off, I will mark it wrong. Like I said, the diversity of this plate is at least 7. But if you're something like 5 to 10, I'll give you points for it. If you're further off, you'll get a penalty. Like if you were to say something like the abundance is 3, I would say no, 1, 2, well, I already told you there's at least 7. All right, know the terms in this lesson, and then work on the worksheet. Let's go ahead and take a look at it. So to lab module 1, type your answers directly in this Word file and then submit the Word file by 11:59 p.m. Saturday, week 1. Procedure 1, what is in the air you breathe? Answer the questions. What is the colony abundance on this plate? Just simply call colonies and give me the colony abundance. What is the colony diversity on this plate? Here you have to divide up the colonies by their different characteristics, like this one and that one and that one. Probably are all the same. They're similar. Obviously, that one's larger and a little bit lighter, but it's very similar. So I'm going to count that as 1, and that would be 2. This would be a 3. So you have to figure out what differences there are and then give me an estimate. If your estimate is too low, like I said, I will mark it wrong, but as long as you're close, I will mark it right. So, well, I'm not going to count them, but I did went through and said, these 3 are probably the same. That would be a little different. And that one's different. So there's at least 1, 2, at least 3, and I'm not going further. You guys can go further. And then procedure 1C, what step would you take to reduce airborne contamination of a sterile plate while you are inoculating it? So don't tell me what you do to reduce contamination before you inoculate it, like always autoclave it and use sterile media. And then don't tell me something after you inoculate it, like I grow it in the Petri dish, taping the plate shut to reduce contamination. That is something you're doing after you're inoculating. Tell me something you can do while you are inoculating it that will reduce the risk of airborne contamination. And to state that specifically, here we have a plate. I take off the lid. It's now open to the air. Anything from the air can fall down from the air, fall onto the plate, and contaminate the plate. So what can we do while we're inoculating the plate to reduce airborne contamination? Procedure 2, what gets on your arms and when you put your arms on the lab bench? Questions to ask is we sampled the table before disinfecting it and then grew what was on the table on the plate. And then we disinfected the plate and then sampled not disinfected the plate, disinfected the table and then sampled the table again. And here we have the growth after disinfection. Answer the question, did table disinfection work? And then procedure 2B, what's the purpose of allowing the table disinfectant to air dry? Or why do we air dry the disinfectant? So answer that question. Procedure 3, what is on your hands? What we do is we take your dirty hand and touch the plate with 3 fingers, 1, 2, 3. And then 1, 2, 3, 4 fingers on this plate. So the right hand and the left hand. Actually, there's 4 here. 1, 2, 3, 4. And then you wash your hands, and then you dry your hands, and you touch the plate again. And here we have the right hand, and there's the left hand. And answer the questions. Does hand washing work? And then question 2, suggest one reason why hand washing did not result in the complete removal of microbes. Hint, it may have to do with the normal microbiota of the skin. If you obtain an answer, you should provide a brief citation of your information source. This question is actually a thinking question, so you will have to put some thought into this one. So when you wash your hand, why does it not result in the complete removal of microbes? Procedure 4, when you kiss your significant other, what exactly are you giving him or her? What's on your lips and what is on your tongue? Here's the picture of your lips. Somebody touched their lips to the plate, and then somebody touched the tongue to the plate. Please note that I put a note there for why this tongue looks like it is a smear right there. It's actually growth right there. And which plate has the higher colony abundance, the lip plate or the tongue plate? And why do you think this is the case, even though the abundance of both body sites is expected to be the same, meaning the lips are right next to the tongue and you probably often lick your lips? What might cause the observed difference in colony abundance? And I'll warn you, you must have 2 reasons for part B here, meaning for full credit, you have to give 2 reasons. And you can easily come up with 3 reasons. I've had very creative students, very bright student, come up with something like 5 or 6 different reasons, but you must give me at least 2. And like I said, 3, coming up with 3 is pretty easy. And then procedure 4C, why was the tongue plate incubated at a higher temperature than the lip plate? Meaning we incubate this plate at a higher temperature than the lip plate. And why do you think we did that? Procedure 5 are microorganisms in the soil. What we do is we take a sample of soil, dilute it in water, and then take the dilution and put it on the plate, shown here. And it says, what is the colony abundance of this soil plate? Is it even possible to accurately answer this question and explain your answer? And then procedure 5B, is there greater abundance of colonies from the soil compared to the lip plate? And I will warn you that it looks like the lip plate is small colonies, but there are some that are very large. And the lip plate looks like it's large colonies. Let me blow that up. But there are some, like right in here, right in there, that are small. So not all of them are large. I mean, those 2 are really small. These 2 there and there, there's 2 in between which are sort of small, but that's really small there and that's really small there. So don't tell me the size of the colonies, because you do have small colonies in the soil plate too. All right, any questions on this lab? If not, I will stop recording.